Thanks very much, and I'm aware that this has been such an exciting meeting that we're continually overrunning, so I'm going to go through this as fast as I can so that you can all get your trains home and get back to where you're meant to be. So I started off thinking of this talk of how stratified medicine and genomics became part of AstraZeneca's DNA from the time that I joined, which is an embarrassingly long time ago, but definitely when that was not the case. And for anybody that's thinking about it, I'm using the term stratified medicine, personalized healthcare interchangeably. There is no particular significance, whether statistical or otherwise, to use of those terms. So just to step back a bit of what we're actually trying to do in the pharmaceutical industry. So about 10 years ago, all pharmaceutical companies were using more or less the same approach. And if you think of somebody like the England football team, and you have a penalty shootout, you can imagine us as all standing, coming up for our terms, taking a shot, using one of these white eggs at the goal, trying to get in as many goals as possible. And to be frank, we weren't very good at it. So you might think, like the England team, that a lot of those shots actually didn't make it to the back of the net. And then not only AstraZeneca, but many other pharmaceutical companies actually changed their strategy because they recognized that that approach was not sustainable. And instead of just having more and more shots on goal, we decided to focus on what would be a really high-quality candidate. So we looked at all of those elements that made a successful chance at getting that ball to the back of the net, whether it was hiring David Beckham or looking at, new sports science or whatever, to actually have that one successful hit. And that's in particular where we, AstraZeneca, introduced a new look at stratified medicine or precision medicine. So what we focused on is, rather than taking just pot shots at this approach, is what actually makes scientific sense and what changes that science, exactly as Tom was telling you, into life-changing medicines for patients all around the world. That was what we focused on. And as part of that, of course, we looked at stratified medicine. So if you go back over a couple of papers that we've published, and John Saville referred to this morning, quite a few of our phase 2 failures turned out to be because we had actually defined the mechanism of action of the drug. So it might be, say, PD-L1, as you've just heard about in immuno-oncology. And then we thought, well, maybe that drug, that population of patients is actually not commercially attractive. So let's go and trial the drug in a much bigger population of patients, which isn't actually related to the mechanism of action. And guess what? A lot of those trials failed. So we've changed our approach, and stratified medicine and the emerging science that we've heard about all over to the exciting meeting today has helped us do that and deliver those better, safer, more efficacious medicines to patients. And what's happened in AstraZeneca is that we've now gone back and we've compared our rates of success all the way from research through to launch of our medicines, and we have actually delivered a 4-fold improvement in productivity. Now, that's still not good enough. It means that if I'm working on a drug in research, it has something under a 20% chance of actually getting to patients in the clinic. It's a lot better than we were doing before. And we are still, of course, looking at ways to improve that. And I'll come back to that later. So, as I said to begin with, stratified medicine, precision medicine, is now in our DNA. And I have personally worked on 4 so-called precision medicine drugs, which we define as being linked to companion diagnostic tests on the FDA website. It's not an easy thing to do. That actually help to match the best patient, the right patient to the best treatment. And because those diagnostics then have to go through a different regulatory procedure, every time you go into a new region and every time you go into a different indication, we actually have 19 linked diagnostics to our AstraZeneca drugs. We don't do this on our own. We are not a diagnostic company. We don't have a diagnostic arm. So we work with diagnostic partners. And currently, we have over 185 million invested in managing those global partnerships. And from where we started, so when we started on this journey, back when I was making the investment case to our senior management for investment in stratified medicine in around sort of 2009, 2010, at that time we had a maximum of 10% of our portfolio following a stratified medicine approach. Now we have 90% of our clinical pipeline following that approach. So a remarkable difference. And I'll come back to describe what we're doing in our genomics strategy, which we're seeing as the next phase of our journey in precision medicine. So some of the things that we have done, and this is not almost by choice, it's some of the things that we have had to do to take our medicines and our companion diagnostics along that journey. And Tom was just talking about our next challenges, about the panel testing. But when we started, we had to, for example, get approval for a BRCA test. And it was the first time that the FDA had ever been faced with a gene that had thousands of mutations, or two genes that had thousands of mutations, and new ones appearing all the time. So they actually had to approve not a single gene mutation, but an algorithm for defining what was a clinically significant variant. Then you've heard a lot about circulating tumor DNA today, and we approved the first drug label that was based on circulating tumor labeling. That came from very early research that we did with a clinical fellow within our labs at Alderley Park, and work that she did, and then has turned into an approach that is used throughout our pipeline and is particularly relevant, as we've heard, for patients with lung cancer. And now we're moving beyond oncology, because we're very concerned that patients in primary care, we heard before, can also benefit from this approach of stratified medicines. And what that means is that they have to have rapid, inexpensive point-of-care testing, and we're working on prototypes for that testing in asthma in particular. So here are just some of the examples there. I won't go through these of what we have done there, particularly with our diagnostic partners, and how we work with, we're able to pick the best partner and the best approach for each drug and for each disease area. Now, I said that we have been measuring our performance in precision medicine since around 2009. So I've been doing this same analysis since we first got our investment in precision medicine, and we've measured the top 10 of our peers on these external measures, these objective measures. And what's interesting, the first time we did this, we weren't even on the graph. We were so far off the graph that nobody had ever heard of us. And now, again, we've got to this position where we are, in some measures, in some metrics, leading, and in other metrics, we are equal ranked with the market leaders. And as I said before, another thing that has changed about pharma is the culture. When I first joined the industry, it was very much about being secretive, keeping things ultra confidential, not having collaborations with anybody. Now we work with academic partners, We have very open relationships with our academic partners all around the world. We work, of course, with diagnostic companies. We even work with other pharma. So Tom and I have a collaboration which is about putting one of our best drugs together with one of his best drugs and exploring that for the benefit of patients. So to move on to the Genomics initiative, this is something very exciting. I'm a geneticist by background, and we announced this back in 2016, our ambition to take advantage of new technology to analyse 2 million genomes over the next 10 years, well, 10 years until 2026. And part of that analysis is to actually physically sequence 500,000 genome samples that come from our own clinical trials. So where we've got information, detailed information, not only on disease state, but also on drug response. And what we're trying to do there is fundamentally to understand the biology of disease. So we know that these approaches have made great breakthroughs, for example, in cancer. But you heard earlier about how we have complex diseases like asthma, COPD, where we still do not know what are the molecular basis, and therefore what can we actually target to make innovative medicines. So understanding the biology of disease, identifying new targets for those medicines, and then it means that as we understand the mechanistic basis of disease, when we go into clinical trials, we have a much better basis, again, to select the best patients for those clinical trials and launch medicines with a diagnostic that can match those patients to the best medicine for them. These are just some of the collaborations that we've set up in the past 2 years for our genomics initiative, and I'm quite sure that there will be more partners as we go along. We cannot do this on our own. And as I've said, this genomic strategy, we believe, is unprecedented in scale and scope. There is something that is distinctive here, even though these large-scale genomics initiatives are being announced almost every week. There's the figure, there's the actual necessity to analyse the 2 million genomes, and we're not doing that because it's a big figure. Let me make clear. We're doing it because our advisors and the population genetics tells us that it is the role of very rare genetic variants that is driving these complex diseases. And you heard some of that analysis, particularly from Magnus, how that is looking in the classic field of pharmacogenetics. And we're beginning to see it as well in the analysis that we're doing. It's obvious that we have to integrate that genomic and clinical data. And going back to the last question, that's not always as simple as it sounds. And as I said, we are building that through multiple partnerships. So here's some of the first results out of that genomic initiative. First of all, on the left here, you can see that there's a reanalysis of the difference that it makes to drug development if you have a genetic association with your drug target. This is an analysis I did myself. It's quite crude. I'm sure as we go forward, we'll be able to make this much more rigorous and much more systematic. But the difference is actually replicating something that we've seen multiple times, and our colleagues in GSK have also published on this. So I think it really is something real. If you have a genetic linkage for your disease, your drug target with a disease in question, those drug targets are much more likely to succeed all the way through from research to launch. It's not a one-to-one relationship, of course, but there are many other reasons why drugs may fail, but it is definitely something that is worth looking at. And then on the right-hand side there, you can see one of the first analyses that we have in chronic kidney disease, and the type of analysis that we are doing, which is a very specialized type of analysis called collapsing analysis with our colleagues in Columbia University, led by Professor David Goldstein. And this allows you to essentially collapse qualifying variants in the implicated genes, so that rather than analyzing billions of genes and polymorphisms and bases throughout the genome, you're actually only analyzing every gene in the exome. And what you can see here is in chronic kidney disease that these genes, which are well-known and validated drivers of familial kidney disease, are indeed the ones that, on a completely hypothesis-free approach, are by far the most significant. And I must say that I haven't seen a p-value since that, like that, in my entire research career. So we do have novel genes, of course, which are now being investigated, which I'm not showing you, and do look forward to seeing some publications on those very soon. So just to highlight our genomics achievements as they stand right now, we've analysed over 240,000 genomes. So one of the pieces of advice that we were being given by our advisory board as we set up this initiative is that it is not so much the ability to do physical sequencing that matters in modern population genomics. The data already exists to a large extent, but what you need to build is the capability to intelligently and rigorously analyze those genomes. So that's what that figure represents, and that's why it is so important. Then we've actually physically sequenced over 12,000 of those clinical trial samples. That figure is going up almost by the day. We're investigating 9 different disease areas of relevance to our therapy areas at the moment. And in that CKD study that I just showed you, we analysed over 400 billion nucleotides to identify those top 405 genes, the proof of concept genes. And at the moment, we have 11 partners in this genomics initiative. Again, this is an external-facing initiative, and we rely on working with many more. So what do you see as the future of stratified medicines? Is it the ability to use big data and all the various data sources and to incorporate them, maybe wirelessly, as we see on the TV, at the point of care when patients like the ones that we've heard about need them most? Is it the ability to have a patient's genome already annotated when they come to see their GP or their specialist for a certain condition, and to be able to pick out what is of clinical significance? Is it to be able to integrate devices and sensors and, again, have all of that data integrated with clinical care? Or is it simply to take patients out of the clinic and out of the hospitals and have them able to enjoy the fresh air, the spring in Cambridge, which is quite beautiful, and to enable all of the things that normally happen to them in hospitals going on behind the scenes while they enjoy more time, more quality of life with their family and with their loved ones.